notification icons and apps. And let's get through this last little bit of stuff. So we're looking at the hormone regulation of oogenesis and the ovarian cycle, otherwise known as the uterine cycle, excuse me, otherwise known as the ovarian and uterine cycle. Then we're going to do the guides when we're done with this. We're looking at an average 28-day uterine cycle coinciding with the beginning of menstruation. Could be the coinciding of a pulsatile release of GnRH at what frequency? 60 to 90 minutes. So when I notice bleeding that first day, I know my hypothalamus is doing this. That'll trigger the release of FSH, that follicle stimulating hormone. So I can awaken a bunch of sleepyhead primordials, about 15 to 20 of them. Why that number, we have no freaking idea. Get your doctorate in it and let me know when you write a paper. But we just happen to know this. And about a day or so later, LH will kind of follow suit. And so by the second day, I have my FSH and LH coming down to my follicle structure. The idea is to add more and more layers of granulose cells around the oocyte, the ova. So at the beginning, with both FSH and LH coming down, I have these low levels of estradiol, not estrogen, coming out of my follicle cells, stimulating other follicle cells to go through mitosis, and that's where I get a larger follicle. Several days later, maybe about the mid-follicular phase, I've lost a significant number of my 15 to 20 already. You may only have 2 or 3 left. You may have 10 left, who knows? But they are not responding, and they're beginning to just simply die off and be absorbed. But now we have a medium level of estradiol, which is now actually getting to circulation, triggering a negative feedback on hypothalamus, which means a negative feedback on GnRH, and a negative feedback on the pituitary, especially the FSH aspect, the cells that create that. Because think of it as taking 242, right? Class started, what, September 23rd, let's say. Can I let students come in on October 23rd? No, they're too far behind the schedule, right? We have to finish this class before we start the next quarter. Well, on this date, I can't awaken any other new primordials. They're too far behind. They're not going to have enough information to survive finals. Finals being the LH surge. So we're telling them, hey, don't send the signal to enroll them into the program. Keep them quiet. Let's use our medium level of estradiol to make sure we don't awaken any other new follicles. Our oocyte, remember, was in prophase 1, and it's going to be kind of chit-chatting to the granulose cells. But now we have this cavity beginning to form called an antrum. Kind of like the colloid in a follicle cell. And this fluid is going to just help a place to create some chemical signals. But the antrum is going to get bigger, the follicle is going to get larger, and we still have our oocyte in prophase 1. A few days later, we're down to usually just one single mature follicle left. Everybody else has not made the mustard, they've died off, they've been absorbed. And we're noticing now 2 new additional hormones being produced. This is probably on a 28-day cycle, maybe day 12, 13-ish in there. Now we have our highest possible level of estradiol, along with now the 2 new hormones, low levels of inhibin. And keep in mind, the hormone levels are important. So I want to ask you to be specific. You can't just say estradiol. You have to say low, medium, high, or whatever it is. You have to say low or high progesterone. We don't have a level for inhibin because it seems to be rather constant in a sense. But now my follicle structure is creating my highest level of estradiol, levels of inhibin, and low levels of progesterone. Now, what is the reason for those hormones? Well, the goal is to create an LH surge. I need a massive release of LH. So how do I do that? Well, I'm going to send lots of signals, hypothalamus, to the GnRH cells. I'm going to get sort of like 2 signals coming in, one from low progesterone, well, I guess on this side, low progesterone and high estradiol. I'm also sending a direct signal via low progesterone right to the pituitary, the anterior, to get another release of LH. And with that high estradiol, low progesterone, and then inhibin kind of tweaking and reducing the FSH levels, that could lead to a massive release of LH known as the LH surge. Now that LH surge can help us do 3 things. is going to help us complete meiosis 1. So now this will talk to the oocyte and tell it, hey, all right, finish the rest of meiosis 1, ramp up meiosis 2, and get up to metaphase of meiosis 2, but hold off one more time. All right, we don't have the right DNA just yet. So freeze at metaphase 2. And follicle, let go of your little oocyte, right? And it'll actually rupture it. It'll some books talk about it as kind of like a pimple popping, right? Literally, it's a rupture, and it just bursts right out. And it's going to leave the ovary and then be swept up by the fimbriae. They're going to create a current of fluid to kind of suck and draw the ova into the infundibulum, and then up into the ampulla as we go through this. Now, the whole purpose of all of this is to reproduce, to create the next generation, to create embryos and fetuses. So we're going to anticipate a fertilization event here. Because how long does an ova live for?
24 hours.
Just 24 hours, right? But now you think about this. An ova lives for 24. How long does the corpus luteum at its shortest lifespan exist for? 10 to 12 days. That's a lot longer than a single ova that hasn't been fertilized. So that's why it's thought of as anticipating a fertilization event. It'll take about 10 days for a fertilized egg to travel all the way down the fallopian tubes and get to the uterine cavity. So I'm anticipating it getting there, creating the chemical signal that's going to begin to burrow into the lining. And how do I know it actually exists? Because this developing structure, is creating a hormone called human chorionic gonadotropin. It says, hey, we actually have a fertilization event. Please don't retire in 10 to 12 days, because if you do, and I happen to burrow into the stratum functionalis, I'm going to be shed right off with that, and I don't want that to happen. So I'm going to give you a signal that tells you to exist for 60 to 90 additional days. And by that time, so 60, that's going to be 3 months, I'm into the fetus stage. So that fetus will basically tell the corpus luteum, okay, I'm here, I'm capable of making my hormone signals. I will talk to my mom's brain and tell her I am here. You can retire and become a corpus albicans. Either way, you become that structure. Corpus albicans, as you can see by the slide, releases no hormones. There's nothing coming from my corpus albicans. Well, we got to get to that next stage here. Because sworn I was on that slide a moment ago. All right, here we go. Push the right button, dude. There we go. All right, there are no arrows coming from this. Now, that was kind of a fast recap of all of this. We want to go back now and sort of look at what's happening with some of our estradiol levels and sort of what's happening with some of our progesterone levels. We're going to want to think about looking at either this chart and also this graph on slide 12. And kind of look at them side by side. So we go to the bottom of slide 12, right? We can see the first day of menstruation, right? And then I know that that is my pulsatile release of GnRH, right? Then by the time I am on, day 5, day 7, somewhere in there where the menstruation stops, we're about the mid-follicular phase, right? Where we're getting a little bit of an increase in estradiol. And what we didn't review and remind you guys of is that with that pulsatile release of GnRH, that led to the LH surge. What is the fastest pulsatile release we have for GnRH? That's every 30 to 60 minutes, right? And if you do the math, that's like 36 times a day, these little pulses of GnRH coming down. That's what helps to trigger that LH surge. Now, I wish I had 3 projectors for today, but I'm looking at my rising levels of estrogen, right, which we noticed, get to the slide 12 here, is going to start happening after we're done with menstruation. To be able to start repairing this tissue, what's the name of the tissue layer that replaces the stratum functionalis? How does it know to do that? It's the rising levels of estrogens in the early follicular phase that tells that layer, hey, start repairing this tissue. Start making some new stratum functionalis. And we can see that with the diagram. I mean, this was actually quite nicely done. All right, I'm beginning to repair. The thickness is getting greater, and it's corresponding to increasing levels of estradiol, so that's nice with that. At the same time, when we have that medium level of estradiol, let's go back to, how do I want to do this? Changing the way in my head how we're going to do this. All right, so let's go to slide 10, because we'll talk about that estradiol. The first thing you want to think about is what I'm asking you guys to do for the feedback mechanisms is to go slide by slide with the low, the medium, and the high, and the medium high, and say what's happening with those various hormone levels. And we said it several times already, just looking at the slide and writing down what that level of hormone's doing. But one thing you don't know about is at medium levels of estradiol, that's going to trigger a thinning, a wateriness of the mucus at the external os. There's going to be a mucus plug at the external os, and we're going to start thinning it out, make it more watery.
That's at medium levels of estradiol?
Because that's going to be about 4 or 5 days before ovulation. Now think about that external os, right? It's at the entrance of the cervix to the cervical canal, then we have the internal os and the uterine cavity. I don't want pathogens getting into the uterine cavity to cause an infection. So nature put a mucus plug there to keep bad things out. Now if it's going to keep bad things out, it might keep wanted sperm out as well if you're trying to reproduce. So that medium level estradiol helps to thin out that mucus. So when women are doing their own calendar clock of their cycles, they're looking for body temp and vaginal mucus stringiness, viscosity, that's what I'm looking for, viscosity. When the viscosity decreases and it gets waterier, I know I'm at my mid-estradiol level. That's my cervix getting ready to allow sperm to pass through there if I'm trying to reproduce. Now with that, we switch over to the idea of the progesterone. We got to do a couple things back and forth here. Again, you're going to go through each slide and look at what low-level progesterone is doing, what high levels of progesterol are doing, and simply write down what their feedback mechanism is. That comes straight from the slides. But there's a few things we have to add to this. So as I look at slide 12, my little black line of increasing levels of progesterone, I notice that they have these little black squiggly lines corresponding to the beginning of the increase of progesterone. Because progesterone is known as sort of the preparatory hormone for the stratum functionalis. These little black lines represent capillaries and blood vessels, the vascular tissue to the stratum functionalis. So as progesterone levels increase, you start to increase the vascularity, the bottom line here, of the endometrium lining. Bring more blood, because we are anticipating pregnancy. So we're sort of like fluffing the pillow, making it really engorged with blood, so there's lots of nutrients. And that's going to be due to increasing levels of progesterone. There's no specific level of it, just we start increasing it after ovulation, we're going to start getting an increase in vascular tissue. It will also trigger an increase in body temp of between 0.3 to 0.5 degrees Fahrenheit. I'll just make it easy, call it 0.5. And I think this graph on slide 12 does that as well. It does, the very top. All right, there's your increase in body temp. Now, women who are trying to do this, measure their own vaginal mucus thickness and measure their body temp, have to be careful about doing body temp. All right? You can't get up, go downstairs, make coffee, get the kids breakfast, and then take your temp. Right? You're going to be hot. Right? So the idea is if you're trying to clock or schedule your own cycle, you have a thermometer on the bedstand next to you, you can simply just reach over and just take your temp. The least activity, the better. If it's up by 0.5 degree, here's the important thing. It tells you that you have ovulated. Not that you're going to. You have ovulated. And it will say increase for the duration of high levels of progesterone. If we notice a drop in temp corresponds to probably, a little pause after my drop in progesterone. Now, we're not quite sure why we have that increase in body temp. It might be to prime the immune system. It might be to increase vascularity to the tissue, really not quite sure. Again, that's things that we've noticed, and we know that it happens, but we don't actually understand why it's actually taking place.
Does that have to be Fahrenheit or Celsius?
It's at Fahrenheit.
That would be a lot.
Celsius would be a little hotter. All right, that would be menopause.
Okay.
All right, now go back to what happened with medium levels of estradiol, right? And look at where medium levels are. Again, go back to this for a second. We'll go to this one, right? The gray box is ovulation, right? We gotta look at this slide, right? There's my medium levels of estradiol. We can look at this slide right here, right? The medium levels of estradiol just before the low progesterone and the high estradiol, which means we're getting ready for an LH surge, which means we're getting ready for ovulation. So we had that medium level estradiol to thin out the mucus, so it's easier for sperm to go up the cervical canal and get into the uterine cavity and find the appropriate fallopian tube. Well, with my increasing levels of progesterone, we are now going to begin to re-thicken that mucus plug. We're going to start sealing the wall again. We want a nice layer of mucus to prevent pathogens from coming in and infecting the developing embryo. Remember, this is anticipation of having had a fertilization event. So when we look at our LH surge on slide 12 with this multifunctional little graph here, I can see the black little squiggly lines corresponding to an increase in progesterone. So I'm increasing the vascularity. I can see the increase in body temp, right, happening right at the edge of my LH surge, and it lasts for the most part through the entire luteal phase. And then right here I can see with my black and blue lines, with the blue lines being estradiol, that the levels of progesterone are beginning to diminish on this graph, right, and the levels of estradiol are beginning to lower. Well, that's because we have now the death of our follicles. Either because it didn't ever get an HCG, so we're on a normal uterine cycle, or we did get the HCG signal, we are pregnant, we confirmed that we're pregnant, but I can only do my job for maximum for 90 days, and I'm now beginning to drop off my progesterone levels. But if that were the case of having HCG, if we did have HCG, and just remember that this would continue at a high level. This is only because we did not get an HCG signal, because we do not have a pregnancy. Don't get that confused with having a pregnancy. Because since I didn't get the HCG signal, my corpus luteum only lives for 10 to 12 days and really begins to die in the 10th and is dead by the 12th. I start having my progesterone levels drop. Well, dropping in progesterone and dropping in estradiol creates a signal for a hormone that you have not thought of in many, 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 many weeks. Prostaglandins. That should sound familiar to you.
Yeah.
How do you remember the name prostaglandins?
They're lipid-soluble hormones.
Too vague.
Icosanoid.
I'll give you a half a point. What did you just say?
Icosanoid.
They are an example of an icosanoid, which is a lipid-soluble hormone. That's why I said memorize the names, but remember their significance. You'll hear them later on in your careers.
Right from cholesterol.
Prostaglandins actually cause the spiral artery to constrict. And if you constrict an artery, you can't deliver blood. If you can't deliver blood, you can't deliver nutrients. So the tissue literally begins to die, which is what triggers the sloughing off of our little cycle here. So the lowering of progesterone, the lowering of estradiol, triggers prostaglandins. They cause vasoconstriction of the spiral arteries. That denies the strand functionalis nutrients. They starve to death, they die, and then we're back to day one. We slough off that dead, dying tissue. And it's the tearing of the tissue off of the endometrial lining that creates the discomfort. And the best analogy I can think of to what it actually looks like, which is kind of a weird scenario, picture, you guys know what a musk ox is? Or a mountain goat that's actually shedding its fur, right? It sheds it in chunks, right? Like your cat when it's really molting, no, not necessarily molting for a cat, but shedding is what I'm thinking of. That's what happens to the uterine lining. It doesn't come off in one nice little sheet. It's actually patches. Like if you go to a really old house and the wallpaper is diminishing on the walls, there's part of the wallpaper still there and parts of it falling off. And it's the falling off that actually tears the spiral artery, which creates the discomfort that comes with menstruation. Now keep in mind the luteal phase is constant, but the follicular phase is the variable part. And just to add another little layer of cool understanding, it's actually this part right here that varies the most. It's some call the sort of the proliferative phase. We're beginning to regrow our sternum functionalis. That seems to be the part that could go for 3 days, 5 days a week, 10 days. Doesn't matter what it is. But that is the idea of one uterine cycle. Pulsatile release of GnRH starting off at 60 to 90 minutes, leading to an increase in pulsatile release of 30 to 60, triggering an LH surge, causing high levels of progesterone, medium levels of estradiol. As those hormones drop off to a certain level, that's my cue to start all over again. So if I had an embryo or a fetus that for some reason had a malfunction, its ability to create its levels of estrogen and progesterones, then the levels in mom's blood will diminish. And you're basically telling mom to do this, which is how you can sometimes get spontaneous abortions. They just simply don't have the proper chemical signal. The body thinks, oh, we're done, and it simply just sheds that lining off. It might happen on day 12, and so you never really knew you were pregnant. You were just late. And you're used to being late because of the stress that we're all under. So you may never know. We won't get into development and pregnancies and like monozygotic, dizygotic twins, fraternal versus identical. But there's an interesting little idea. How many of you are left-handed? All right. So supposedly, 20 to 30% of you had a twin in utero that disappeared.
Sorry, the left-handed people?
Left-handed people. Yes. Interesting. Not confirmed just yet, but it's a high probability that left-handed people actually had. It's called the vanishing twin scenario, where women have gone for 3D ultrasounds and they found twins or triplets. Go back a couple of weeks later, and there's a pocket where the amniotic sac used to be, but there's nobody in there anymore. It just disappeared. And those that were born tend to be left-handed. So they're going, wow, wait a minute. So if you're left-handed and there's only one of you, there might have been another one of you that was there, but for some reason has now totally gone. We don't know why that's true. Certainly isn't the fetus killing the other one off. That's for great white sharks and nurse sharks for embryophagy. All right, this makes sense somewhat, okay? Now, yeah, this idea here. Let's get off to the males. So you want to think about the anatomy of the testes, right? The seminiferous tubule lined by Sertoli cells, and then outside on the periphery are these interstitial cells or cells of Leydig. Now I put two different ways of thinking about this up here. This is a little nice chart that shows us the organization of our pathway for the development of sperm. I have my spermatogonium, which is our stem cell. It's along the periphery of my seminiferous tubule. It's going to go through mitosis, right? And we're going to create our primary spermatocytes because of that. Once we have gotten a primary spermatocyte, then it's a non-stop sequential series of meiosis 1, meiosis 2 to create our spermatids. There's no interruption, there's no pause. Speaking of pause, we didn't remind ourselves of what causes the ova to finish metaphase 2.
Oh,
I remember it was fertilization, but then how do we define fertilization? It was the sperm DNA joining up with the ova DNA. That is the fertilization event. That's what triggers the rest of our meiosis process. So sperm DNA joining up with egg DNA. Now back to the sperms. The spermatogonium goes to mitosis, so I can always maintain new stem cells, so that's why they never get old, in a sense, compared to oocytes. But then we have the uninterrupted pattern of meiosis 1 to meiosis 2, and we're going to get these spermatids that are down here. So from 1 to 4, right, we're looking at spermatogenesis. But I need to take my 1950s rocket ships, right, and make them more like cruise missiles down here. I have to change their morphology. So the first thing to talk about is, how do we go about signaling this ability to initiate spermatogenesis? And this is the first diagram of 2 that shows us the hormonal regulation of spermatogenesis. We have hypothalamus and a release of GnRH, which they didn't show as a pulsatile fashion, but it is a pulsatile fashion. Goes to pituitary, LH, FSH, just like females. I mean, nothing here is different between males and females. I get my gonadotropins released, and then it's simply my last chance to have you guys think about looking at the diagram and going literally with what the diagram is going to tell me. Well, there's LH, and where is it going? Well, it's going to interstitial cells. I can see that. And what does that cause them to do? Well, to make testosterone, because there's my little black arrow. And what does testosterone do? Well, it takes a little practice, but you realize, oh, they're saying leads to secondary effects like maybe deepening of voice, body hair, muscle development. So those are your secondary sex characteristics. But we're more interested in actually spermatogenesis. So testosterone from the interstitial cells goes into this purple thing, which is a Sertoli cell. And see how my spermatocytes are not in the Sertoli, they're between these 2 neighboring ones, and there's your little tight junctions. But the testosterone will diffuse into the Sertoli cell, through the cells, and get into the lumen, where I'll have my developing sperm cells. And it'll attach to something called APP, whatever the heck that is. Well, let's figure out what it is by going backwards. APP apparently is the androgen binding protein. Oh, so that's like the thyroid binding protein for the bloodstream. It allows me to sequester and hang on to and build up the level of testosterone in the lumen where my developing sperms are going to be. Okay, well, where did that androgen binding, because testosterone's an androgen, where did that protein come? How did it know to make that protein? Oh, it came from the cell products. Well, whose cell products? That's the Sertoli cells. How do I know that? Because it says so right there. So now I can go forwards now that I work my way backwards. Sertoli cells create cell products, one of which is the androgen binding protein that goes into the lumen to attach to testosterone. Well, what else do Sertoli cells make? Oh, they help to trigger the developing sperm, which we knew about from a previous day. But also, oh, they're the ones that create the inhibin. And what does inhibin do? Well, it goes up out of the seminiferous tubule into the bloodstream to do what inhibin always does, which is to inhibit FSH. Well, let's look at the last thing that testosterone does, and we'll go through it all over one more time. Testosterone, besides creating for secondary sex characteristics, besides triggering Sertoli cells to help with developing sperm that'll bind to the androgen binding proteins in the lumen, but it also does negative feedback on LH and FSH. So it's sort of like the male's version of low levels of progesterone, but now in a negative feedback pattern rather than a stimulation pattern. And the level of inhibin corresponds to the level of sperm production. But that's one way of looking at it. Let's look at it in a different way from this one, from your author of the McKinley textbook. We can have a little more light because it's a more brighter image. And again, practice looking at it. Go, okay, now they're showing me the pulsatile arrows. That's nice. goes to the pituitary, the anterior one, triggering LH and FSH. And where do these hormones go? Well, this one's going to go to this, whatever this thing is. Oh, that's a sustentacular cell. That's a Sertoli cell. All right, that matches this idea, right, of FSH going to a Sertoli cell. And then the LH will go to, be careful, follow it all the way through, not to a Sertoli cell, but to these guys on the outside edge of your seminiferous tubule. That's these guys. Those are the interstitial cells of Leydig. All right, well then these guys, right, what are these guys doing? Oh, they're making a green line apparently. They're making a number 3, which they're making testosterone. Okay, and that's what this thing says, right? Interstitial cells make testosterone. Testosterone then can be secondary sex characteristics, right? Go up and inhibit, right, GnRH and the antipituitary, and also trigger spermatogenesis, right? But now here, right, there's no direct mention. You got to come back to their FSH path. FSH going to sustentacular cells. They will then create inhibin, and inhibin will definitely inhibit FSH. There's no real proof of this, so you are to scratch that out, right? Get rid of this red dotted line right here until otherwise proven. He's the only one that does that, so we're not going to believe him for the moment until there's more proof. But I realize that sustentacular cells make inhibin, and inhibin will do inhibiting FSH, which is what this one shows. Right? And I trust Silverthorne more than I do Martini. Plus, my other textbooks agree with Silverthorne and not Martini or McKinley here. And then sustentacular cells, besides making inhibin, they will also make the androgen binding protein, which attaches to testosterone. So this number 3 in the green arrow is another way of saying this right down here. So I can hold on to testosterone, let the levels increase, and together, which neither one of these two really say, is that you need both testosterone and FSH going into the Sertoli cell to trigger sperm production. You need both testosterone and FSH to trigger the production of sperm. It isn't just FSH. You need both testosterone and FSH to trigger Sertoli cells to tell the spermatocytes to get off their butts and get going. Now we can do a comparison between spermatogenesis, right, and oogenesis. In terms of their stem cells, remember that females have all their stem cells in place before they're born. Right? Males don't start creating sperm until they reach sexual maturity. Females, when they go through their rounds of meiosis, right, will create these things called polar bodies. And the polar bodies are the ejected DNA as a result of that meiotic phase with the least amount of cytoplasm as possible. It's like an escape pod. And all that's in there is the DNA, the complement DNA of each of those chromosomes. So if I'm separating homologous pairs, one set of them are going to go into my polar bodies. And that cell will go through meiosis, and then this cell will go through round 2 of meiosis, so that cell becomes 2. There's 1, there's 2, there's bless you. So I'm all done. I'm going to have 1, 2, 3, 4. Those are my 4 cells from meiosis. You're going to get 3 polar bodies and 1 actual haploid gamete. In males, right, we go through uninterrupted. We don't create any polar bodies. There's no ejection of DNA. I'll still get 1, 2, 3, 4 haploid cells, my gametes, that we will then call spermatids. To get to be a fully formed sperm, I have to go through spermiogenesis. So spermatogenesis was the creation of primary spermatocytes up to these immature, morphologically not proper spermatids. These are the 1950s rocket ship ideas. The idea then of, whoa, we're missing a slide. Holy crap, okay. We will add that to this little comment here. To go from the spermatids, the 1950s rocket ships, to the cruise missile idea, is you have to get rid of all unnecessary organelles. So all of the rough ER, smooth ER, Golgi apparatuses get taken out. Excess cytoplasm gets taken out. And that's what they're trying to show you here, which I don't know what happened to that slide. That's interesting. That's what they're doing here, right? See how much more slim and trim these guys are, right? All the unnecessary organelles are gone. The cytoplasm has been reduced, and that is what is known as spermiogenesis, the maturation of spermatids to sperm cells. You know, it says spermatids becoming sperm cells.
Is sperm a bacterial cell?
Huh?
Is sperm considered like a bacterial cell or a
Bacterial cell?
prokaryotic or a eukaryotic?
I don't know. Where did it come from?
Doesn't matter.
Is there a nucleus in a sperm cell? Okay. Did it have organelles?
It did have organelles.
That means it has to be a prokaryote. I'm sorry, you're a good guy. You may do that. Eukaryote. It has to be a eukaryote. Oh, wait a minute. We can't go from eukaryote to prokaryote.
It's alive.
It's alive, yeah, that's a thing. It's a virus. It's not dead or alive. Okay.
Oh, man.
You guys are done.
That's it.
That's it. All right? Nothing else. It's enough as it is. Okay? So, everybody listen up for a second before you get too rowdy on me. Listen up. All right, Wednesday, what happens on Wednesday? Your lab final, right? So keep your eyes up for an email in terms of the timing. There's a class having their final before us. They have to break down. I have to swoop in and set up. So I'll get a better feel after talking to the instructor about how we can coordinate this. We may start as late as 4:30, but be prepared for a 4 o'clock start time for the first lab group. Second lab group, we will start at 6:30 for your lab final. It'll give you a half hour to get your bearings and kind of chill out from the traffic rush here. Okay? I can make a lecture if you want. No, there's no lecture on Wednesday, right? That'll give you almost an entire friggin' week, right? So pace yourself. After the lab final, you're allowed a party that night. That is it. The next day, lay out your final review in terms of allocation of time. Review material versus new material. Do each day. I will be here this next Saturday as well. It's our last Saturday. If you have questions, come. Otherwise, those of you that haven't had lab yet, I will see you in lab. See the rest of you on Wednesday.